this is a very specific story about cell movement and whatever, but it's actually a much more general thought. The thought is that what's happening here is that by exporting your stress, you are facilitating alignment and cooperation because your problem becomes everybody else's problem, right? It's a lot easier to keep identity among the different cells when you're stressed out. I don't care, I'm fine. Then there's you and there's me, and there's a separation. But if your concerns become my concerns, it's not because of altruism. It's just you're stressing me out, and so I'm motivated to I'm motivated to help you because it's a you don't need a separate altruism mechanism for that. So then the problems, they're shared problems basically, and shared problems, much like shared memories, are a key to getting everybody working towards the same outcome. So that's a kind of cognitive glue because it binds everybody to the same purpose. And
Michael Levin is a biologist at Tufts University, where he heads a lab focused on developmental biology, cognitive science, and bioengineering. Today, we'll discuss his paper, Stress Sharing as Cognitive Glue for Collective Intelligences, a computational model of stress as a coordinator for morphogenesis. Please subscribe, and I hope you enjoy our fifth conversation together. Mike Levin, thanks for coming on.
yeah, good to see you again.
Yes, such a pleasure. It's always a pleasure to have you on. Thank you again. Thanks. And well, let's see. Last time we discussed your paper on memory having agential properties, and I'll actually publish that today. Actually, I think this morning. So yeah, and so I'd love to talk about one of your recent papers that you co-authored, called Stress Sharing as Cognitive Glue for Collective Intelligences, a computational model of stress as a coordinator for morphogenesis. And if you wouldn't mind, could you give us like the high-level summary of the paper and then tell us about the experiments that you ran for it?
Sure. Yeah. So this was work with Lakshman Shrisha, who's a very talented student, and working with us on these kinds of issues. So I think first let's go back to the fundamental notion of the cognitive glue. This idea of the cognitive glue. Yeah. So the idea of the cognitive glue refers to the set of properties and policies by which individual subunits interact together to give rise to some sort of emergent collective that has some property that the individuals don't have. So either it can store memories that the individuals don't have, or it pursues larger goals in different spaces than the subunits can. And so there are many different kinds of cognitive glue. We've been studying certain kinds having to do with memory anonymization and hackability and some other things. But in this paper, we test in a computational simulation. We test one particular hypothesis, and the hypothesis that I had was that one way to augment the emergence of higher level minds out of lower level subunits is by enabling them to share stress. And so let's then back up and talk about why stress is important in the first place. So the idea is that something that's fundamental to cognition of any kind is a kind of a homeostatic error minimization scheme. That is, you have preferred states, you remember what they are, you measure the current state. If it doesn't match, you take actions to get you closer to that preferred state. And then when the error is within some allowable tolerance, then you stop. And so this is a very simple kind of homeostatic loop that's kind of at the basis of a lot of things. And we've been studying the idea that morphogenesis, so the self organization of shape during embryonic development, during regeneration, during cancer suppression, during resistance to aging, all of those things, is a kind of anatomical homeostasis. That is, you've got a bunch of cells. The cells are part of a collective, so they have a kind of collective intelligence. And that allows them to navigate this anatomical morphospace, and it allows them to navigate the space of possible anatomical shapes and get from where they start, which initially might be a single cell egg, or it might be a damaged body of some kind, but to reduce that error and to get to the target morphology that they want, the complex shape. And individual cells have no idea what that shape is. They're solving little tiny physiological and metabolic goals and transcriptional goals, but the collective is able to remember these large scale goals and pursue them. And the reason I call it specifically a collective intelligence is that it's not just complexity; it's really the ability to reach those goals despite all kinds of novel circumstances. So things change, and if you've got the ingenuity, some degree of ability to navigate that space and still get where you want to be despite novel circumstances, that's a kind of intelligence. Okay. Mm-hmm. So the reason stress is important is because one way to motivate action is to so you've got this homeostatic loop. You've taken a measurement, and there's a delta between what you have now, and that might be your size or the shape or the number of fingers you have or something like that. You've taken a measurement, and it doesn't match what you're supposed to be. Okay, so there's an error. So now you have to motivate the cells to actually now spring into action and reduce that error and get you where you need to go. So one way to thinking about stress in this context is as a system-wide parameter that indicates how far are you from where you want to be. So you're under stress to the extent, and this is not a new concept. I mean, it's been studied right in physiology for a really long time, but now I'm extending it to anatomical homeostasis, and the idea that the stress is what motivates you to try to reduce that delta. So then one can imagine the following scenario. Imagine a variety of cells, and this is one I think it was figure 1 in the paper. That you've got a bunch of cells, and this is just one example. There's a million ways this can play out, but just imagine that there's a bunch of cells, and there's a cell on the bottom that's in the wrong position, and it needs to get to the top, and there's a bunch of cells in between. Now, the cells in between are where they're supposed to be. Their stress is very low. When your stress is low, you're unlikely to make changes. You're not motivated to make changes. Life is good. Everything's fine. But the cell below is unhappy. It wants to try to go up. So as long as the stress is very localized, it's very hard for it to go up because the other cells are not motivated to help. They're fine where they are. Mm-hmm. So one way, if you think about evolution, one easy way to facilitate cooperation and alignment towards goals is to let the stress be leaky. So let's say that stress down below is that the cell down below is very stressed out, and there are internal molecules that represent that. So various kinds of stress molecules, and we're actually studying those now in the lab. We have a project on that. But what you can do is you can make them leaky and have the cell sort of let them either actively or passively let it secrete outwards. And then what happens is that because these stress molecules are universal, the cells nearby are now receiving those stress molecules, and now they're stressed out because their stress level is now measurably higher. They can't tell, much like with the memory anonymization thing, they can't tell that these stress molecules don't belong to them. Right, the symbol is what it is, the molecule is what it is, and so now their stress goes up. Now, when their stress goes up, everybody's a little more plastic, right? If you don't think you're quite where you need to be, then your willingness to make changes, that threshold to do something goes up. They get a little more plastic. They start to move around a little more, and now the cell that's at the beginning can now move upwards, right, and move through them, and then everybody's stress will go down. This is a very specific story about cell movement and whatever, but it's actually a much more general thought. The thought is that what's happening here is that by exporting your stress, you are facilitating alignment and cooperation because your problem becomes everybody else's problem, right? It's a lot easier to keep identity among the different cells when you're stressed out. I don't care, I'm fine. Then there's you and there's me, and there's a separation. But if your concerns become my concerns, it's not because of altruism. It's just you're stressing me out, and so I'm motivated to. I'm motivated to help you because it's you don't need a separate altruism mechanism for that. So then the problems, they're shared problems basically, and shared problems, much like shared memories, are a key to getting everybody working towards the same outcome. So that's a kind of cognitive glue because it binds everybody to the same purpose. And you can think about it too, like when we look at an early embryo, let's say, like an amniote, like a mammal or a bird or something, where it's a blastodisc. You've got, I don't know, 50,000 cells, and we look at that and we say, there's an embryo. Well, what are we counting when we say there's one embryo, right? There's lots of cells. What is there one of? Well, what there's one of is a story that all of the cells are committed to about where they need to go. They're aligned physically because of planar cell polarity. They're actually like physically aligned, but more importantly, if they're aligned in anatomical space with respect to the goal that they're going to try to reach. And then if you do perturbations, if you intervene in various ways, they will do their best to still get to where they're going and overcome actually a wide range of things that would otherwise cause birth defects and so on. Okay, so that's kind of the background to this. So we were interested to ask a simple question: this idea that shared stress would improve morphogenesis, that it would be evolutionarily, it would be an attractive kind of feature. That's what this paper was designed to test.
Cool. Yeah, thank you for sharing. So I have a million questions to ask you. One is about how the cells can't tell the difference between internal versus external stress. What is it that's special about stress that they can't make that distinction?
It's actually not special about stress at all. It's that no molecule really has any kind of metadata on it as far as we know about where it came from. And this is a story that I worked on a few years ago too with regards to memory anonymization. So the gap junctions, right? The amazing thing about these electrical synapses that connect cells in the body is that when small molecules that represent specific, let's say specific memories of events that happen, so some calcium spike corresponding to an injury or something, when that propagates to the neighboring cell, that cell has no way of knowing, was that me that got poked or was that my neighbor? Because the memory engram is what it is, and this links back to our previous talk, right? As you try to interpret your memories, you have these molecular traces within you that are the result of past events, and you don't have access to the ground truth of what happened. You only have access to these memories. So if you have a memory of there was some kind of damage, you don't really have anything to compare that against. That's your referent now. Well, then I guess I've been damaged. You have to interpret these signals. Right now, I'm obviously greatly oversimplifying because there's a million different kinds of molecules and whatnot. But the idea there is that once we've established what the different symbols mean, whether that's stress or damage or nutrients or whatever, once we've established what these things are, once we come across that in our internal milieu, it's ours now. It's very hard to distinguish from false implanted false memories from what really happened. So that I think is the same thing with the stress. The cell is no, I don't think the cell can tell, wait a minute, that's not my stress, that's coming from somewhere else. Other than there may be other metrics, and if one is off the charts but everything else is cool, you might have this like so-called cognitive dissonance where you're trying to figure out, is there really a problem or isn't there? I'm getting mixed signals. So there's some complexity there, but fundamentally, right, if we all know that this molecule is a sign of DNA damage or a protein misfolding or some other larger kind of stress, which is what we're talking about here, once you've come across it, that's your conclusion.
And where does the stress originate from, say, within, say, one cell? Like, is there stress sharing within the cell itself?
That's an excellent question, because of course there are sub-modules within cells, and how it is that different gene regulatory networks and different organelles and different aspects of the machinery, how do they react to it is very interesting, and I'm not sure that much is known. For example, I think it would be an incredibly interesting project, and we're doing something like that, and Martin Picard and other folks working on mitochondria are probably looking into this. But the passage of stress from a really a pretty self-contained internal agent like the mitochondria out to the outside of the cell itself, right? Our stress molecules propagate it that way too. Can the mitochondria achieve some form of cooperation with the cell by the same method? It's an interesting hypothesis. I don't know. I don't think we know about that yet. At least I don't know.
Yeah, interesting. And I'd love to dive into, because you gave a great overview of kind of like, I guess, the setup and a lot of the general concepts that led to the experiments within the paper. But could you tell us about the specific, and does this fall under artificial life? Actually, I was wondering okay.
Yeah, I think this is an example of artificial life. Specifically, there's in silico embryogeny, which is this idea of trying to mimic some kind of self-organization, some sort of embryonic development, and link that to evolution, and ways to simulate all of that. And it's also artificial life because, on the one hand, we're very interested in the details of how this works in the biological world, because I'd like to use this for therapeutics and for regenerative medicine and so on. But in another sense, this is way broader than that, because we're interested in life as it can be, to paraphrase or quote Chris Langton. So not just the way that life on Earth happens to do it right now, and how it evolved, but actually all agents that are made of matter that are going to have to be this kind of multi-scale organization, where you have to get your parts to cooperate towards some sort of larger scale outcome. Is that going to be universal? I mean, I actually think I think stress is an incredibly powerful generic concept. There's all the way from, the most basic version is geometric frustration. Is that familiar?
I'm not familiar with that now.
It's like imagine, there's a couple ways to do it. Imagine tiling the plane. So you've got a plane, and you're gonna put down little shapes, and they all have to fill the plane contiguously. Well, there's certain scenarios where you start tiling, and then eventually you run yourself into a corner where you can't put down a shape that makes everybody in the neighborhood happy. So it's like this idea that somewhere there's going to be a discontinuity somewhere. There's going to be some mismatch somewhere, and you can try to minimize it, right? But that idea, and this is captured in things like icing models and models of magnetic domains trying to all align. But sometimes you can't have a situation where everybody aligns perfectly, and there's this like mismatch, and it causes a stress. But overall, there's a notion of temperature where, and it can be actual temperature, or it can be a kind of a symbolic temperature. But the idea is that when the temperature's high, everything's plastic and changing. But as you slowly cool, everything has to settle down. And that notion of stress, the minimization of mismatch of local scenarios, is trying to produce a global optimum. And sometimes it finds it, and sometimes it finds a good scenario that's not actually a global optimum. But that kind of thing is super common in physics systems, in different kinds of geometries, and other kinds of math. So I think, and this is one of those kind of weird sounding and controversial things, but I think it's all one spectrum. I think geometric frustration is actually frustration, in the same sense, not the same degree, but the same sense in which cognitive scientists or psychologists might use the word, because we too are a collection of modules that have different goals. We hope they are all aligned. Sometimes they're not. And frustration in cognitive systems, I think, comes from the same very fundamental dynamics of trying to get the pieces aligned given constraints, and oftentimes cannot be, you can't make everybody happy, because life is complex and there's all these complicated requirements and limitations. So
oh, interesting, fascinating. Yeah, are you familiar at all with parkour?
Yeah, I mean, I've seen it.
You see it? Yeah. No, I'm not asking, like, do you do it on the weekends or something? Like, actually, I'm a pro, amateur parkour. But it makes me think of, well, sometimes you see clips of these guys, they're out of their minds, but they jump from, like, 2-story artifices, like, onto the beach, like onto sand. And, they do it carefully. You need a lot of practice to do it properly, but they land, like, on their toes, and then they displace the energy into their heel, and then it goes all through their body until it dissipates, and they roll out of it, right? And I think about that a lot with this and stress sharing, in that, like, there's tons of stress on their feet, and then they dissipate that across their entire bodies, right? It kind of gets spread out, and by doing that, they don't break a bone or something like that. But like, you need to be able to share and dissipate the stress in a way. And
that's a very cool point, which I actually had not appreciated before, and nor is it mentioned in that paper. This idea that, I mean, in our model, the stress sharing is all very practical and useful. So you're sharing stress because you want these cells to do something. But I think you just pointed out something interesting, which I need to think about more, which is that you might just want a reservoir that doesn't do anything. It's not that, right? It's a reservoir into which you can dissipate stress that isn't necessarily for anything other than giving extra degrees of freedom to allow the really important things to align the way they want, and then it's fine if the reservoir is all disordered, right? I think that's really interesting. I hadn't thought of that before, and I wonder if there is an analogy like that in biology. We should think about it. I wonder if there are both in anatomical space, but also in physiological and transcriptional space, if there are these like regions of disorder that are there to soak up the extra stress that themselves aren't really used for anything. That's kind of cool.
Cool. Oh, I'm glad I shared it. Awesome.
Yeah.
And so for the and for the paper, and I know it's probably going to help, with your permission, if I could overlay some of the visuals from the paper, because you had things like the boxes and neighboring cells like sharing, but you had, let's see, three groups, let's say, three test groups, one that shared stress, one that didn't share stress, one that was hardwired to achieve this goal of like a smiley face in a grid space. Could you share with us, like, I mean, what did you learn from the experiments, and just what came out of it?
So two basic things to establish. First, the reason for the smiley face, and we now have, I think, 3 or 4 papers with a smiley face in it. And the reason is simply because that smiley face looks like the bioelectric pre-pattern of the early frog face development. So this was a paper that Danny Adams and Laura Vandermerg and my group worked on years ago. And what they discovered was something we called the electric face, which is a pattern of distribution of bioelectric potentials in the early cells of the embryo that are going to later become the face. And what this pattern is it serves as the memory of what a correct face is supposed to look like. So remember that in order to do this homeostatic loop, you have to actually store the set point, right? Like your thermostat has to know what the right temperature range is in order to reduce and the same thing for temperature and hunger level and pH and everything else in the body. In morphogenesis, you can't just store a number or a range the way that we would for hunger or pH. You have to store a pattern. Like here's what a correct face looks like, and if we're deviated, we need to work hard until we get there, and then we can stop. So those patterns aren't super detailed. They're kind of rough outlines. The details are worked out later by the cells locally, but the rough outlines of the body plan and of the organ structures and positions are laid out by these pre-patterns. And there are chemical pre-patterns like morphogen gradients and things that people have studied in the past. And the thing that we study are these bioelectric pattern memories, and the electric face is one of those. So it literally tells the embryo where to turn on the genes that are going to be important for having retina and in the eye. So here's where the eyes go, and there's 2 of them, and then here's where the mouth goes, and then so it controls the gene expression. So that's why the smiley face is that this is a rough model of the kind of thing you see in frog face, vertebrate face development. The other thing that's important to explain is the notion between hardwired and competent embryos. In lots of rough descriptions of how evolution works, you have this idea of we have the genome, we have the genotype, and then you mutate. The mutation operator works on the genotype, and then that genotype encodes some set of features. Those features are evaluated in the environment, and then we call that selection. And that's the phenotype, is like how well do you actually do in the environment, and then that's what gets selected for or not, and there's your cycle. So the important thing to realize is that that simple description is missing one really important feature, which is that between the genotype and the phenotype lies a really important layer of physiological intelligence, meaning that your genotype in most cases, I mean, there's a few things like enzymes and things like that, but by and large, your genotype does not directly code for any of these large scale features. Okay, there's no, if you look at the genome, it doesn't say how many eyes you're supposed to have or how many bones in your forearm they're supposed to be. You're not going to read any of that out of your genome directly. What the genome encodes are physical and computational properties of the subunits, the proteins that cells get to have. So that's the hardware. The genome encodes the hardware. Once that hardware is up and running during development, the cells use that hardware to recall all kinds of memories, to self-organize patterns, to make decisions, and to do this kind of context sensitive behavior to solve various kinds of problems. So what the genome actually encodes is a problem solving agent with preferences, with memories, with goals. And that agent will navigate in physiological space and anatomical space to give rise to the final phenotype. And that's what then gets selected. So that level, and then we have lots of other papers talking about that level of problem solving competency in between the genome, the genotype and the phenotype is really important, because it makes all of evolution work differently. And we can talk about what that is. But in that paper, when we said hardwired, what it means is that those embryos don't have that middle layer. They literally just go, here are the genes, and the genes just tell you what you do, and that's that. So, across the spectrum of living systems that we know of, there's a whole kind of a continuum of different places of how plastic are you. So, for example, nematodes, right? So, like C. elegans are probably the furthest on the left of that spectrum. They are very cookie cutter. So every C. elegans has exactly the same number of cells. They're all numbered. They all come from these lineage maps. There's as far as we know, and we have to be very humble about this because this is what we think. We may not have tested it sufficiently, but as far as we know, there are very few extemporaneous creative decisions being made. It just kind of does what it does, and that's it. It's a very hardwired program kind of thing. On the opposite end of that, you have planaria. I mean, planaria are just unbelievably plastic. They can accommodate an incredible number of new scenarios, and they still do what they need to do. And they're very, very plastic. You can't really, their development mode is very chaotic. I mean, they're just incredibly plastic and responsive, and they solve all kinds of problems. And then in between, you have everything else. So you've got mammals. The adults aren't super regenerative, most of them, but like as embryos, you can cut them into pieces and get twins and triplets and so on. And you've got some salamanders that can respawn and regenerate and solve some interesting problems. But anyway, so there's a whole spectrum of those. But that's why it's important. So in that paper, we show here's how this plays out in these kind of hardwired systems. Here's how it plays out in the competent systems. So the ones that have that layer of problem solving in between. And then we tested this idea. So let's see. So you asked what did we find? Okay, so what we found just overall, we found a few things. Including stress sharing, because in the model we can turn it on or off, right? That's the nice thing about these in silico models, is you can say what does it look like when you don't allow them to share stress versus when you do. What we found is that allowing them to share their stress improves the morphogenetic efficiency of the collectives. The individual embryos do better, and populations in which you allow stress sharing within the individual, they reach their anatomical targets faster. So faster and more reliably. Now, we do not have, in this model, we don't have stress sharing between embryos. That's a whole other interesting thing. And we have some work on embryos communicating with each other to help them solve problems. We haven't linked that experimentally yet with the whole stress story. But it's the same person, Angela Tung, who's working on this amazing stress project. And this was her, the multi-embryo communication was also her work. So these things will come together. But yeah, so they do better. They reach their goals faster. The population does better. One thing, as predicted, that we measured is that allowing them to share stress enhances individual cells' movement. So it makes it easier for them to move, and that makes sense given the story I told you before. We didn't bake that in, by the way. All of these things that I'm telling you now are emergent in the sense that surprising and not directly baked into the code. We didn't push them to do any of these things. These are the things that we observed after the fact. There's actually pretty minimal assumptions in the model, and so they move further, and their radius of influence grows. So because they're allowed to share stress, what we see is that actually their ability to change other cells' behavior expands out. So this is not obvious. You might think that, well, you just said that you let the stress expand outward. So of course, right? But that doesn't have to be the case, because you can imagine in some scenarios that I sort of diffuse stress outwards, but that doesn't mean that I have any influence on what the other cells do. right? We don't know. They could ignore it completely, right?
Right, right.
We have no idea what they're
Hmm.
actually going to do.
Hmm.
Yeah. But it turns out that yes, it actually in this model, sharing your stress does allow you to influence pretty far off cells. So that means that we are getting this like integrated, the level of integration between the components is rising, right? So we saw that, and another interesting thing that we saw was that we couldn't we couldn't infer or guess the anatomical states that they're trying to reach from observation of these dynamics. In other words, if you didn't know that there was a smiley face, right? Which the smiley face, that bioelectric pre-pattern that we did put in because they have to have a set point, an anatomical set point that they're working towards.
Sure.
But if you didn't, but if you were observing this from the outside and you didn't know that that's what they were doing, you couldn't read that, for example, by looking at the patterns of stress. In other words, there's not an obvious one-to-one mapping between, because you might think that, oh, if you set up a stress pattern that looks like the face, then the cells will just kind of come, get it. So it turns out that's not what happened. I kind of suspected that might happen, and so we looked, but actually that didn't. And there's an interesting parallel here, which is much like with neural decoding. If you're thinking really hard of I don't know, of a daisy or something, if I read the signals coming from your brain in whatever modality, what I'm not going to see is a little picture of a daisy, right? It's a very indirect mapping. I mean, people are working on how to infer what it is that you're thinking of from the electrical signals, but it's certainly not because you have a little daisy pattern hanging around in your brain. So this is kind of like that. It's extremely indirect. The stress patterns, and there are all kinds of patterns, but the patterns that are there don't directly allow an external observer to know where this is going and why. Like what's the goal? But the system itself does, right? From the inside, from the perspective of each embryo, the stress pattern is exactly pointing to the thing you need to do, but the external observer doesn't see it. So that's kind of cool. It's you see what I mean? It's a primitive version of something that's sometimes called privacy in the philosophy of mind literature, which is that it's very easy for you to decode your own neural net and know what your memories mean. Well, it's not trivial, but it's a lot easier than it is for me. So we have to, right? We have to go through all this extra work, and it's still not very good, to try and find out what is this brain thinking about. It's really hard from outside, but from inside, most of the time under normal circumstances, you have a pretty good decoding of your own the mapping between the physical state and the mental state is pretty is pretty tight from inside. So I think that's interesting. It's a very minimal model of this distinction of what it's like to be a system and experience things from the inside versus to try to guess what's going on as an external observer.
Yeah, it's fascinating. Yeah, it's like not intuitive at all. You'd figure you could see it from the top down looking in, but it's like you need to be within the system to understand what's happening.
Well,
Um,
that's and I think that's a key aspect of what we mean by the spectrum of agency. So just
mm,
imagine you've got a landscape and it's sort of bumpy, right? You got this like bumpy landscape, and there's a bowling ball on the landscape. If you want to know what the bowling ball is going to do, your perspective on it as a third person external observer really tells the whole story, right?
mm,
You don't need to take the perspective of the bowling ball. You can look at it from the outside, add up the
mm,
forces. You know everything that's going to happen. But if you've got a mouse on that landscape, good luck. You can't
mm,
just do that. You actually have to take some degree of perspective of the mouse and ask, what do I know about this landscape? Did I get rewarded and punished in previous days? Am I hungry? What do I see? What is my mind capable of perceiving about this landscape? What senses do I have? Like, it becomes for high agency systems, it becomes really important to be able to take their inner perspective. And in fact, you
mm.
can define sort of the degree of the inner perspective that a system has to be the degree to which an observer would have to mind meld with it in order to know what's going to happen next. Or
Mm.
that's the predictive action, but how about the interaction, to have a productive interaction with it, right? So whether that be to control it in the case of simple systems, or to be friends with it, or to have some sort of spiritual interaction with it in the case of like far on the spectrum of high agency systems. In all of those cases, what you really need to know is how much inner perspective is there, and how much of that do I need to take that perspective in order to have a high efficiency interaction that enriches us both.
Very cool.
And so I think in this model we're seeing a very minimal, this is nowhere near a mouse, but it's a very minimal model of that kind of idea, that here's something that is extremely functionally salient from the first person perspective of the system, but is very hard to extract information from a third person. And so it doesn't take much. You don't need a big fancy brain or anything like that for this property to show up. It shows up even in pretty minimal systems.
Wow, so much. It's fascinating. I'd love to get some context too on what occurs for the cells that don't share any stress information. So how do they communicate? Like what's their strategy for achieving the smiley face?
Yeah, so in models like this, what you have is the ability of cells to have certain behaviors. So maybe they pursue specific gradients, maybe they get other kinds of signals that have nothing to do with stress. And then the whole embryo gets evolutionarily rewarded for having the right pattern, because if you don't make a correct face, then your fitness will be low, and then that whatever that genome, right? So what the genome encodes are the low level rules. Here's what the cells can respond to. Here's what they can. Here's how they can act. Whatever. And so the rules that don't allow the cells to do what they need to do to create a face, those eventually get weeded out. But even under those circumstances, what we found is that not being able to share stress makes it much harder to do this. And actually that's actually an important segue into kind of the evolutionary implications of this. So the reality, and this is something else, another paper that that we had some years back, Danny Adams and Laura Vanderberg and we had this paper on craniofacial fixing. What happens is that if you artificially make, we call these Picasso tadpoles, if you make a tadpole where everything starts off in the wrong position, so the mouth is on top of the head, the eyes are off to the side, like it's like a Mr. Potato Head where everything is kind of scrambled. What happens is that you actually get a pretty normal frog out of it at the end, because all of these things will move around in novel ways to still get you the pattern that you need. That right there is the morphogenetic competency that I talked about. It's not just the fact that every organ moves a certain distance, because you might think that's how you could build a tadpole, because all tadpoles look the same and all frogs look the same. Maybe all you need to remember is, okay, the eyes go forward a little bit and the jaws come out, and that's it. You just remember how each piece goes. But if you do that and you start with a scrambled position, then you're going to end with a scrambled frog, because everything will take the normal amount of steps from the wrong position and it'll end up in the wrong position. But that's not at all what happens. That's what would happen if the system was hardwired. But it turns out, and everybody thought that's what people thought up until we did these experiments, where it turns out that that's actually not hardwired at all. And all of these organ primordia will move in novel paths. In fact, sometimes they go too far and have to double back, but they all move around until you get a normal frog face, and then they stop. So that's that competency. And the implications for evolution is, right, so just think about this. Imagine that you had a mutation, and the mutation moves the mouth. So most mutations do more than one thing. They have more than one effect in the body, right? So you have some kind of mutation, and let's say it does something really fantastic somewhere, in the tail or in the gut or something, but it also results in the mouth being moved off kilter. If you had a hardwired system, that animal wouldn't be able to eat. It would die, and you would lose that mutation from the pool, and you would have to wait until you discovered an independent way to both have that nice whatever the good effect was, and keep the mouth where it needs to be. And who knows if there even is such a mutation, right? Because most things affect most other things. There may not even be one that does that cleanly. And so that's right. So that would really make evolution take a really long time for those kinds of things. Instead, what you have is this developmental competency that if you do end up with the mouth in the wrong location, that's all right. It'll fix itself. Give it a week, and it'll get back to where it needs to be, and then you're good. Now evolution has the ability to explore the positive consequences of that other mutation. So the competency, that error repair kind of mechanism, allows all kinds of mutations that would have been fatal or deleterious otherwise to be either neutral or actually beneficial, because it takes care of everything that can be fixed. It fixes it. And this is and so here's the implications for evolution. Imagine that a tadpole comes up for selection and it looks really good. Like all the stuff is in the right place. It's eating, it's doing, everything's great. What selection doesn't know is it really good because it had an amazing genome that put everything where it needs to go? Or is it really good because, well, the genome was kind of so-so, but then it fixed everything? Selection has no way of knowing that, because all selection sees at the end is the phenotype.
Right.
And so by having this competency, you're hiding information from selection. You're shielding the genome from selection. Really now can't tell, right? Is my structural information pretty bad, but I have amazing competency, or is my structural information spot on? It can't distinguish those. So it makes it harder to do the thing that it's supposed to do, which is to improve the genome, right? It makes it harder to do that. Now, when you do that, something interesting happens, and we have a couple of other papers looking at the same phenomenon. One by Lex Fridman, that ask the question: well, if evolution can't be as good at selecting the great genomes, where does it spend all of its effort? Like what is it doing at that point? And it turns out that what it's doing is improving the competency. If it can't see the actual genomes, all the effort ends up going into improving the competency. Well, if you improve the competency, then the problem gets even worse, because now it fixes even more things, and now there's even less of the genome that you can see. And so it becomes this like self-reinforcing spiral, where the more and so then evolution cranks the competence even further, which hides more of the gene. So you get into this spiral. And I think all in all, I think that spiral is basically an intelligence ratchet, because what it does is it pushes for tissue, which I've called an agential material, right? It pushes for the material of life that evolution is evolving over to be higher and higher competency at solving new problems it hasn't seen before. And that initially that starts in physiological and transcriptional space, but eventually in anatomical space, and eventually that busts out into three-dimensional space of conventional behavior, so what people recognize as intelligence, and then who knows, linguistic space or something else beyond that. But I think that's the fundamental drive, is like that early dynamic is a positive feedback loop that cranks this very basic intelligence up and up through the different problem spaces.
Interesting. Is it related to how, because I noticed one thing from the paper, I'm not sure if this is like the genotype-phenotype relationship or for some other reason, but there's a quote from the paper. It said the benefit of stress sharing is especially noted in the later stages of evolution, when mutations cannot be relied upon to solve the problem efficiently and more precise cell movements are required. Is that what relates to what you just said, or is it
does? It does, because the more complex the more complex the scenario you're trying to push, the harder it is to micromanage that at the lower levels. So it's the same reason that it's the same reason for in regenerative medicine, I've been pushing on this idea of, rather than trying to rewire the genetics or the protein structure, we should be going more top down and really taking advantage of the competency of the cells and tissues, and communicating novel goals to them, and letting the system take care of all the downstream, all the complex downstream machinery to implement it. It's harder, and the more complex you get, the harder and harder it is to get there by micromanagement. And that's we're well aware of this in neuroscience, in the sense that if you have a rat and you want it to do a specific little circus trick, if you wanted to run the muscles like a puppet, in theory you could, but that would be extremely difficult, right? Tracing back all of the signals that you would have to give to the muscles of the feet, going up through the spinal cord, all that stuff, that would be really hard. Or you can just train the rat. And the reason that that's so much easier, it requires less information, it requires less effort. What it does require you to do is to recognize that you're dealing with a problem-solving agent, and to understand a little bit about its mind and its rewards and punishments, currency, and what kinds of things it can learn. But what you don't need to know is the molecular biology of the neural structures. And this is why humans were able to train dogs and horses for thousands of years, knowing zero about what's between their ears, because they offer you this incredible learning interface that you can take advantage of. And that's the thing. When you want to do complex things, it's really hard. But notice that what I think is super cool is that the way and it's really bioelectric from beginning to end. The way that the physiology is working is that while it's super hard for us to know, hey, so which synapses do I need to tune exactly for you to like, for the rat to ride the little bicycle or whatever? That's a really hard question from a third person external observer point of view. But for the rat itself, having been given some behavior shaping cues and some rewards and punishments, the system does all of that. What does it do? Well, it tunes all the synapses and puts the neurotransmitters where they need to be, and reinforces some neural connections and blocks off some others. All of those crazy complex steps are actually handled in the body of the organism. We couldn't do it, and we don't need to do it. But past that interface, the system itself does it. So I think that again goes back to this thing where it's quite different to be doing it from the perspective of the system itself.
Mm. And how does let's see, I want to try to narrow down the questions here for our remaining time left, but is there a balance between how much information sharing is optimal between the cells, or how much stress sharing is between the cells? Like, can too much information be counterproductive?
Yeah, for sure. And ours was an extremely minimal, kind of simple
Mm.
system. The reality is very, very nuanced because, yeah, absolutely, you can flood the whole system with stress where everybody's just freaking out, but there's no gradient about what anybody can do to make things better. So that's obviously not good. And also, there are multiple different types of stress indicator molecules that are used, and I'm sure we only know a few of them. But that again is probably is going to be implementing some sort of much higher specificity system where I can get, where you can put out more complex systems that more complex signals that will get this set of cells stressed out, but not this other set of cells, right? So you can actually fine tune that whole thing, and no doubt evolution is making use of that. But we didn't model any of that, right? In our system, it's just very simple, and yes, you can go to you can turn the gain too high for sure.
Is there, well, I guess for this experiment specifically, because I imagine more broadly there is, but a relationship to, I believe it's mentioned in the paper, stigmergy. So for the audience that doesn't know, it's the ability of subunits like cells or ants to leave each other messages in an external medium of the environment. So is there like for this experiment itself, was there an environment, or was just the environment the other cells? Like, does that make sense, the distinction
Yeah,
there?
yeah. We didn't really, right. We didn't really put a lot of effort into simulating things like extracellular matrix and
Okay.
other cells that are not involved in the morphogenesis, which in real life there would be. We didn't spend a lot of effort on any of that. But you're right. In the real situation, that absolutely would be the case, and that could provide all sorts of scenarios. For example, if you leave some, and we don't know yet if this is something that actually happens in the biology, but certainly could, that if you leave some of those stress molecules around in the matrix, some other cell coming into that, you might be long gone, but some other cell coming into that area would say, oh yeah, this is where bad stuff happened, so you could certainly imagine something like that. And it's stigmergy, I think, also in the broader sense, which is another way to think about certain kinds of stigmergy is you offloading some of your cognitive load to others and using others as a memory scratch pad. So to the extent that I can reliably convince somebody in my environment to do something, I don't have to think about it. I don't have to worry about it. The fact that they're doing it is the memory. In fact, I can forget all about it, perhaps. So this ability of to any time that you have a causally effective signal that you're sending where you're pretty sure what effect it's going to have, you can now start to use all of the receivers as part of your own mind. They're part of your memory they're part of your memory system. And it's like Andy Clark's work on the expanded mind, right? Is that all of the tools in your environment and other agents, which are also parts of your, high agency beings in your environment, they are also part of this global, this global mind that goes beyond what a single human is doing, and certainly beyond what a single cell is doing even.
Oh, cool. I have to. What was the name of that person?
Andy Clark.
Andy Clark. I'm not familiar with his work. Okay. Yeah, I'll
Oh
check
yeah,
that out.
it's really great stuff. Yeah, he's in the UK. I'll send you some links
Oh, thank you.
for the show notes. But yeah, it's very good.
Yeah,
And
yeah.
yeah, it really, he really talks about this idea of exporting, whether that be through writing, just a simple notepad or an iPad or some kind of a network or a communications channel or friends or whatever it is, the ability to offload some of your cognitive structures to other things outside your skull. And I think that's a very powerful notion. And we saw this to some extent too, is that the kind of the radius of influence of these cells when they share stress becomes much broader. So now you can ask another interesting question: is imagine that you're a third person observer that comes in and you want to like reverse engineer the system. You don't know what it does, but you want to reverse engineer it. So you're watching the cells do things and the signals going back and forth. And what you want to do is draw a boundary around the natural units of, like, what are the subunits here? Is it like you can imagine aliens coming down to Earth and saying, okay, so should we be looking at the ecosystem, the individual animals, the cells within the animals, the tissues, the molecules? Maybe we should be looking at molecules. Like, where do you draw the boundary between what are the natural places to draw boundaries? So prior to stress sharing, a really natural place to draw those boundaries would be around individual cells, because they're both basically functioning as autonomous units. And there's some math that's been developed around looking at signals and asking, to what extent can we say that these signals are these states are causally linked, right? Is there some connection between them? Well, so before stress sharing, you would naturally say that it's the individual cells, but after stress sharing, it becomes actually much harder to say where the boundaries of the agents are, because they're so connected and functionally, causally, they're functioning together as a unit to achieve certain things, that you might actually be tempted to draw your boundaries around some sort of, I don't know, it's actually, we haven't actually done this, but maybe we should. There may be some sort of isopotential areas in terms of stress or something like that, where if the aliens were to draw a boundary around individual humans, what that would mean is that they said that all of the neurons that are in other cells that are sitting in our brain, those are not the individual subunits. They all come together, and let's draw a boundary around this whole thing. This is where the action is, right? But they could have picked out the individual cells. In fact, they could have picked out the individual projections of the neurons, or little domains on the cell membrane or something like that. But the question is, like, where do you draw? And that's something that Larissa Albantakis gave a talk in our group recently, talking about her work on IIT, and that's one of the things that theory is supposed to be able to answer. It's a mathematical theory that says, okay, there's a room with a bunch of living organisms in it. How many, right? Like, how many subunits are in here? It's a very non-obvious question.
Oh, I gotta check out that talk too. Yeah, I love. Very interested in IIT, so that's one that I'll share that I'll look it up and share this. Share
Yeah,
that in the description of this
it's on
video.
our channel. She gave a great talk.
Oh, it's on your channel. Cool, cool. I'll check that out. And one of the last questions I want to leave you with, if we could zoom out a little bit and think more broadly, and you can use, I guess, reference this paper, or I mean, all of the work that you've done related to stress. How does this research impact, like, your personal day-to-day view of stress? So the stress we have just as people, does it impact? Is there any crossover? Can you share any, like, I
Yeah.
guess, high-level views about it?
Well, one just funny thing is, this was, I don't know, months ago, but when I was talking to my dad, and I said to him, I described the hypothesis in the paper and everything else, and he said, let me get let me guess, you made a whole theory, focused around stress. Big surprise. He said, it's because you're stressed out all the time. That's why you have a model that's all about stress. Yeah, I don't know. I think that just in general, trying to keep an eye on the flow of indicators of stress. So in our model, there's a very specific, stress molecule that gets separated. But I think in our lives, we are often stressed out in various ways by various triggers, and we often don't recognize what those are or how we're interpreting them. And so I think, if anything, I've become a little more conscious about trying to be more deliberate and about asking, okay, clearly this thing is a stressor. Why? What does it indicate, if anything? Because maybe it has a good role and it's indicating that you're deviating from some sort of goal state that you want it to be in, but maybe it's pathological and you're misinterpreting it. And I mean, this has been said a lot long before, that in the past, certain kinds of adrenaline meant that you had to run fast to get away from a predator. Nowadays, we get that same exact adrenaline when you get when your boss yells at you, or you get a nasty reviewer report or something for your paper. But it's inappropriate because there's nowhere to run in physical space. That's not what this is for. So just being more deliberate about how you respond to these stressors and can we find them, I think is something that bears keeping in mind.
Yeah, cool. Yeah, thank you for sharing. And then the last question I'll ask you is, what's the future of this research look like? What do you have coming up next for it?
Yeah. Yeah, I think for this, the next big step is going to be experimental. So we have a whole project looking at actually being able to because we know what some of these molecules are in biology, so we can track them. And so you can do some really interesting work asking about what are the different stress patterns within tissue under certain circumstances. And we're talking birth defects, aging, cancer, degenerative disease, all these scenarios, regeneration. What is the pattern of stress? Where does it propagate? To what extent can you manipulate and address those patterns as a more subtle way of getting towards repair and things like that? And to what extent do they induce repair? To what extent do they block repair? How much inability to regenerate is because the stress is not being handled in the right way, or in fact not generated, or something we don't know. But yeah, we have that's basically right now at the bench. We're gonna do a whole bunch of work. Hopefully in this coming year, it'll be published, and then we're gonna go back and do more computational modeling with everything that we've learned to now try to make some very specific models that we can then use to try to infer therapeutics. So what are the uses of anxiolytics? What are the uses of certain kinds of other neuroactive, let's just say, neuroactive compounds that would manipulate stress both in neural and non-neural systems. So it's the bench work that's coming next.
Wow, so exciting. Very cool. We'll have to keep an eye on that space. And wow, this is a great conversation, Michael. Thank you so much. I think we got so much out of this, and I just appreciate your time so much. And
awesome. I appreciate that. Thanks for having me on. Yeah.
Yeah, thank you again. And yes, stay well. Maybe we can talk again about the AI. I think that was one thing. Maybe we could get this trio of conversations together. And yes, we'll have a wonderful day. And thank you again.
Yeah, thanks very much, Carlos. See you soon.
Awesome. Thanks,
Thank
Mike.
you.